All right, guys, let's talk about the immune system. It is very complicated, so we are just going to do a brief overview of the immune system. So let's start with the cells of the immune system. So before you get to this cell, you have something called a hematopoietic stem cell. You've seen it written as HSC or HPC. Okay, it is CD34 positive. CD stands for clusters of differentiation, and all that is an antigen on top of the cells. It's used for identification. Now, once you get to this stem cell, this is called a multipotent stem cell. Okay, and then you have two choices. You can go to a myeloid progenitor or a lymphoid progenitor. Okay, let's talk about the myeloid. Now, there are three differentiations of this myeloid progenitor. First of all, it can go to what you call megakaryoblast. Okay, and these produce megakaryocytes, which are platelets. Now, these first two are not shown on here. Now, the second one is an erythroblast, and these turn into erythrocytes, which are red cells. The last one is it forms what they call a myeloblast. This in turn turns into granulocytes, which is the neutrophil, eosinophil, basophil, and mast cell, and the monocytes, these agranular cells, okay, which are the macrophages and dendritic cells. Now, right here, the mast cell is actually a basophil in the tissue, and a macrophage is a tissue monocyte. Okay. Now, these dendritic cells, okay, they are antigen, what they call antigen presenting cells. So are the macrophages, and we're going to talk about that in the next slide. Alrighty? Okay. Now, let's talk about the cells of the lymphoid cell system. So you have lymphocytes, or it's a lymphoblast. Then it turns into the lymphocytes, and you have your B cell lymphocyte, and you have your T cell, and then your natural killer cells. So the B cell progenitor cell they have plasma cells and then memory cells. These plasma cells will turn into antibodies. Okay. Now, your T cell progenitor cells, you have T helper and T cytotoxic cells. Right? The T helper cells, they are CD4 positive, and they stimulate the B lymphocytes. The T cytotoxic cells are CD8 positive, and they release chemicals that kill viruses and cancer cells. Okay, now these guys, in order to do this, the T helper and T cytotoxic, they have to have the antigen presented to them. And again, we're going to talk about that in the next slide. Okay, now this last group, these natural killer cells, they have a faster kill response to viruses and cancer cells, and they don't need the antigen to be presented to them. It's like a little killing machine. Okay, now all these cells are produced in the bone marrow, but the T cells, okay, they mature in the thymus. All right, now just as a little FYI, on a bone marrow transplant, okay, they used to go in and pull the bone marrow from either the sternum or the hip, and you can appreciate that that's doesn't feel very good because they have to do it about 100 times because you only get about 10 mils of bone marrow from each stick and they need about 1,000 mils. Okay, so what they do is you do have these progenitor cells hanging around in the peripheral blood. So they inject the donor with something called a GCSF, which is a granulocyte colony stimulating factor, and it's what's known as a cytokine. Okay. After they inject this, then they harvest the CD34 positive cells. Remember, those are your hematopoietic stem cells. Okay. Donor blood is then put through a filter, and after the CD34 positive cells are removed, the blood is returned to the donor. Okay. Antigen presentation. Okay. Now they're using a dendritic cell. Okay. So this is for and what they call an exogenous antigen. It's found outside the body. So these antigens are either inhaled, ingested, okay, or injected. Now, y'all have heard of the MHC, the major histocompatibility complex. In this case right here, okay, this is for a T helper cell. So you need an MHC class 2 on here. All right. And so what happens is the phagocyte eats the bacteria, the antigen goes to the surface, and it presents it to a T helper cell. So the red part is the antigen, and then this little guy right here is the TCR, the T cell receptor. Okay, you got to have one. Now, with T helper cells, you need the MHC class 2. Okay, and the MHC class 1 and 2, those have to do with recognition of self from non-self, which is a basic tenet of immunology. You have to be able to tell the difference between self and non-self, okay? And that's what the MHC does. Now, if you're looking at endogenous antigens, and we're talking about viral proteins in any affected cell, okay? They're degraded and displayed on the surface of the cell. Now, for these T cytotoxic cells, you need MHC class 1. Okay? And the antigen presentation can be done by a dendritic cell or a macrophage. Okay, but basically it eats it, presents it on the surface. The helper cell and the T cytotoxic both have this T cell receptor, and then they have the antigen. So the helper needs the MHC class 2, and the T cytotoxic needs MHC class 1. Talk about immunoglobulins. IgA, found in mucus, saliva, tears, and breast milk, and protects against pathogens, especially in body cavities. Okay, and so this is called a dimer. So it's two. Here's your IgG. It's two of these linked, and then in here, in this area right here, it's called a J chain, and it connects two or more of these basic units. Okay, IgD is part of the B cell receptor, activates basophils and mast cells. IgE protects against parasitic worms. It's responsible for allergic reactions, and it's found in the membranes of basophils. The IgG is secreted by plasma cells in the blood. It's able to cross the placenta into the fetus. It's involved in the anamnestic or secondary immune response. And this is a good measure of the strength of your immune system, that you develop that anamnestic response. Okay, and the last one we have is the IgM. Okay, responsible for the early stages of immunity, so they're involved in the primary immune response. Okay, they also have a J chain right here. Okay, and remember that links the basic units. Okay, and this is the IgG is an example of a basic unit. the complement cascade. Okay, so there are three basic functions of complement. One is chemotaxis. Sorry, that's the process of calling immunocompetent cells to the site of injury. Okay. Lysis is when they destroy them, and opsonization is when they coat a bacterium and enhance the phagocytic process. Okay, so basically, this is the classical pathway, and then it goes into the alternative pathway. So I just wanted to show you the different stages and everything, but the main thing, okay, is that at the end, it forms the MAC, or the membrane attack complex, which lyses the cells. Okay, so the C5b, C6, C7, and C8 punch a hole in the cell, and C9 just destroys it. All right, now the majority of the complement components are synthesized in the liver. And that's all. Thank you.